Hello dear students, welcome back again to Chinmoy's YouTube channel. So this is the second part of respiratory system where we are going to discuss about few experiments on gaseous exchange. Let us start with today's episode of experiments on gaseous exchange. Now at the end of this lesson, what we are going to learn? We will learn about that the green plants can produce carbon dioxide during respiration. The first experiment we will prove that. In the second experiment we will see the carbon dioxide is produced by germinating seeds. In the third experiment we will see that oxygen is used up in respiration. In the fourth experiment we will see that carbon dioxide is produced during respiration in germinating seeds. And last but not the least, we will see that the heat is evolved during respiration. So these 5 experiments we are going to deal today. Now let us start with the first experiment. Now here we can see in this diagram, now let us see this diagram. We have taken a bell jar. So this is the bell jar and these are 2 conical flasks. Okay, we have taken an air pump, that is it is not actually here, we have put a air suction pump over here and we have put some soda lime water and a glass sheet. Now we have placed the bell jar on a glass sheet and connected the glass jar with 2 conical flasks, one here and one here. So 2 conical flasks A and B are connected. Now we have shown you that the 2 conical flasks containing lime water, as we have shown in the figure. Now we have placed a potted plant inside the bell jar. So it's a potted plant, it has been placed inside the bell jar. We have sealed all the connections with Vaseline so as to make these airtight. So all the connections here are sealed with Vaseline to make it airtight. Now this experiment should always be performed in dark so that we can see that no photosynthesis is happening. So we have covered the bell jar with a black paper or a black cloth to prevent the photosynthesis so that any carbon dioxide produced during respiration is not used up during photosynthesis. Okay, now we have started pumping outside air into the apparatus with the help of a air suction pump. Okay, so air suction pump, what is happening? Through when it is coming, the air passes through the soda lime water and the soda lime absorbs any carbon dioxide present in the incoming air. Now let us see it in the flask A. When the air passes through the lime water, flask A, the lime water does not turn milky. But when this air again passes through the bell jar, having a potted plant inside, now having a potted plant inside, we have seen that then when this air is again passing through the flask B, this lime water turns milky. Why does this happen? So what is the observation? Observation is here, when the first air is pumped inside, the lime water didn't turn milky. So here, what is the use of this experiment? That carbon dioxide is produced during respiration. So obviously it is produced because when the air is passing on through this soda lime water, this is turning milky. So hence it is proved that the green plants produce carbon dioxide during respiration. So this is the first activity or first experiment of today, and it is proved by this simple experiment that carbon dioxide is produced during respiration from this potted plant. Now please learn this diagram with all the specifications very clearly, because in your ICSE what you will get this diagram, you will get to write what are the observations, what is the inference, and what are the things happening during the experiment. So they will give you a picture, they will ask you that what is this experiment showing. So name the things, name the process happening. So you should learn each and every diagram very carefully. Now let us move on to the next experiment. So here the next experiment, what is this experiment about? That carbon dioxide is produced by the germinating seeds during respiration. So here we have taken 4 conical flasks or glass bottles and arranged them in order. So from flask A, where we have placed soda lime, and in flasks B and D we have placed lime water. And what we have placed in flask C? In flask C we have placed 8 to 10 germinating seeds. Now what we have started doing, we have started drawing air from the delivery tubes in flask A towards the flask D. So what do we observe? We will observe that the lime water turns milky in that D conical flask. Why that is happening? In the B conical flask, the lime water doesn't turn milky because there in the first, the soda lime water, the soda lime is there, so carbon dioxide is absorbed if any carbon dioxide was present in the air. But as soon as it is going to the C germinating seeds bottle, then what is happening? Carbon dioxide is produced by the germinating seeds, and when this passes through the D or the flask D conical flask, then it turns the lime water milky. So what is the inference? The inference is that this experimental setup will show that carbon dioxide is produced by the germinating seeds. So I think this is clear to you all. So from flask A to flask D, we have told you that here as the carbon dioxide is absorbed here, so no carbon dioxide is present here. But here as the carbon dioxide is produced by the germinating seeds, when this air is passing through this, it turns this lime water milky in this D flask. So it should be clear that carbon dioxide is produced by the germinating seeds. Now let us move on to the third one. So the third experiment shows that oxygen is used up in respiration. Now here what we have done, we have taken two flasks. Flask A and flask B. Now in flask A there is germinating seed, and in the flask B we have kept the boiled bean seeds. Now on the wet cotton both are put, and a beaker and a small tube containing lime water or soda lime has been kept inside the conical flask. Now what is happening in flask A? In flask A we have placed germinating seeds, and in flask B we have placed boiled seeds on wet cotton. Now we have suspended a small test tube containing soda lime, a mixture of slake lime and sodium hydroxide in each flask. Now the soda lime absorbs any carbon dioxide released by the seeds, and we have connected each flask with a water containing beaker down. So this is a water containing beaker, and we have connected it to the water containing beaker. Now we have noted the level of water in the delivery tube connected to the, see here the mark is here. If you can see this mark is here, you will find that the delivery tube A, it is gradually showing a greater rise in water level. Why? Because oxygen is being used up. So it takes oxygen from this water. So water level is rising up, and if we introduce a burning paper into each flask, what we will see after removing the corks, we will find that the flame is immediately put off in flask A, but it continues to burn for a while in flask B. So what is this showing? That here it doesn't burn, it puts off, and while in the other one it is burning for some time. So what is it showing? That this shows that there is no oxygen in flask A, which is being used up by the germinating seeds, while in flask B there is no respiration occurred due to dead seeds, and so a little amount of oxygen was present in flask B, which was not yet used up. So this flask B, the level of water is reaching here, and as it is not used by the seeds, it is stagnant here only. But gradually here we can see the oxygen level, the water level rises because there is a demand of oxygen in flask A. So what does this experiment prove? It is clear, we can draw an inference from this experiment that the oxygen is used up during respiration. So as we know that the germinating seeds are respiring and the dead bean seeds are not respiring, it is used up in flask A but not in flask B. And it is very much clear from this experiment that oxygen is always used in respiration. Now let us move on to the fourth experiment. So what is the fourth experiment? Now the fourth experiment here is to show that the carbon dioxide is produced during respiration in germinating seeds. Now here also we have taken two flasks, flask A and flask B, and so we have kept soaked bean seeds, boiled bean seeds, and wet cotton wool and carbolic acid. So these are all the ingredients or the requirements of this experiment. Now what we have done, we have placed soaked bean seeds on wet cotton wool in flask A, and in the flask B we have put boiled bean seeds on wet cotton wool. Now we have put few drops of carbolic acid in flask B to prevent any bacterial growth on dead seed. So carbolic acid is preventing the bacterial growth on dead seeds. And now we have corked each flask and placed them in well ventilated room with adequate light temperature for few days. Both the flasks have been kept tightly corked and in adequate light and temperature for few days. After a few days, you will observe that the seeds in flask A have germinated, but there is no sign of germination in flask B. Now this test, we have tested the gases. Test the gases in each flask by removing the cork and subjecting them to containing the lime water. So just we open the cork and we hold the test tube containing lime water. So in flask B we will see that the lime water doesn't turn milky, and flask A the lime water turns milky. This shows that flask A contains carbon dioxide produced by germinating seeds, and the gas produced in flask B shows no effect on lime water, showing no carbon dioxide was released by the dead seeds. Now from this we can draw an inference that observation of this experiment is that carbon dioxide is always produced during respiration in germinating seeds, and hence from this turning of lime water milky, we can show that in flask A germination of seed has happened. In flask B, where we have put boiled bean seeds, no germination has happened. So it is very clear with this experiment. If you are having till now any doubts, any queries, please ask me in my comment box so that I can clear it up with all your doubts and queries. Now we will move on to the last activity of today, that is how the heat is evolved during respiration we will prove. Now the experimental setup to show that heat is liberated during respiration, how we will show, what are the things we have taken in this experiment. We have taken two thermos flasks A and B and about 200 seeds soaked in water up for about 24 hours. We have kept boiled half soaked seeds and washed them in dilute carbolic acid to prevent any bacterial growth, as we have done in the previous one. And we have placed live soaked germinating seeds in the flask while dead seeds in flask B. We have inserted thermometer in each flask and we have sealed the mouth of each vessel with cotton wool. Okay, so what does the observation show? After 3 to 4 hours, the temperature in each flask we have to check. What we will find that the temperature in flask B does not show any rise in temperature, while the thermometer in flask A shows a higher reading. Why? Because germinating seeds are here, and we can see that germinating seeds always give out heat during respiration. So experimental setup for this is to prove that heat is liberated during respiration. So these are all the 5 major experiments for gaseous exchange during respiration in plants. So I think it is all clear to you all. If you are having till now any doubts, any queries, go through your textbook, go through my video again and again, and don't forget to subscribe to keep on getting the updates. Okay. So thank you for today, and I'll be posting the differences between respiration combustion and respiration and photosynthesis in the next video. Okay, thank you.